Hello, so today we'll be going over human immunity and applications of immune response. I'll be going over cell immunity and its communication. So the immune system is made up of a variety of different cells that work together to fight an infection. Different white blood cells, also called leukocytes, are used depending on the immunity response. There's two types of immunity. So innate is nonspecific. It's the first line of response. They have immediate effects, and cells included are macrophages, NK cells, dendritic cells, mast cells, neutrophils, basophils, and eosinophils. Adaptive is specific. It's a second line of response. It has long-term effects. There's humoral and cellular immunity, and there's the T and B cells are primarily used within this immunity. The difference between the two is that innate does not target a specific type of pathogen. It kind of kills all foreign invaders within the bloodstream, but an adaptive immunity that kicks in when the specific virus is detected and it targets that. So I'll be going over the granulocytes that are part of the innate immunity. So neutrophils are the first responders. They send signals, kills pathogens by ingesting through phagocytosis, similar to macrophages. Eosinophils help fight infections and parasites and cause inflammation. And basophils release histamine during an allergic reaction with the help of mast cells. Additional cells within the innate immunity are monocytes. Once an infection is detected, it becomes a macrophage and engulfs pathogens. And NK cells are a type of lymphocyte. They kill viruses and recognize them for future reference. The cells a part of the adaptive immunity are lymphocytes, which are found in lymph tissue. B cells present antigens to T cells and produce antibodies. T cells help kill infected cells and signal assistance to other cells. Cell communication. There's many types of processes to communicate, like through cell-to-cell contact, receptors, or signals. Cytokines tell cells how to fight an infection, and chemokines inform cells where an infection is. So the types of cell signaling, there's autocrine, which is when a cell sends a signal to itself. Paracrine is when signals are transferred locally between nearby cells. Endocrine is long-distance signaling by hormones through the bloodstream. And direct contact is communication between gap junctions and surfaces.
Hello, so my topic is going to be pattern recognition receptors. And what are PRRs, which is obviously short for pattern recognition receptors? And PRRs are proteins on or inside immune cells that help your body detect pathogens like bacteria, viruses, fungi, or cell damage. And you can think of them as the body's security sensors. They detect danger and alert your immune system. And why are PRRs important? They are important because they are the first line of defense against infection. They help distinguish self from non-self, and they play a role in inflammation and autoimmune diseases if overactivated. And then I just made like a chart so you can see toll-like receptors, which could be TLR. They are found in the cell surface and endosomes, and what they detect is bacteria, viruses, and fungi. Nod-like receptors, NLR, they are found in the cytoplasm and they detect bacteria components. Rake-like receptors, RLR, cytoplasm, they detect viral RNA. C-type lectin receptors, they are found on the cell surface, and they detect fungal carbohydrates, sorry. And then here's just a little picture of um, what it would look like. And then what happens after PRRs detect something? The activation of immune signaling, PRR.
Hi guys, my name is Wyatt. I was tasked with going through the complement system and the Christian worldview aspect and how it pertains to microbiology. First, we're going to go over the complement system. The complement system is a series of inactive proteins that are found in the blood and other bodily fluids and the tissues of the body. This system consists of many different proteins, with 9 of them being considered the major proteins that can split into fragments of the main protein. An example of this is going to be the C3 protein that splits into C3A and C3B. This system works on adaptive immune deficient defenses, as they have specialized functions that help them remove and destroy the invader bacteria. The complement system is activated in 3 different ways. All 3 ways lead to the formation of the C3 protein. The first way is going to be the alternative pathway, which is triggered when the C3b protein binds to the foreign cell surface and then allows other proteins to bind to the C3b. These bonds eventually form the C3 convertase. If you notice in the picture that the C3b is both a product and a trigger, this is possible because the C3 protein is unstable and it's susceptible to being broke down. So this allows the loop of C3b being created, but also using C3b to create C3b. The second pathway is going to be the lectin pathway, which occurs when mannose binding lectin, or MBL, binds to mannose. Mannose is a monosaccharide that's found on the surface of bacteria and fungi. This bond allows the formation of C3 converse or convertase. The third pathway is the classical pathway. This path is triggered when antibodies bind to an antigen. And this interaction is between antibodies and the antigen. Whenever they interact, the components of the complement system can be seen kind of similar to the lectin pathway, and they also form T3. So there's 3 different outcomes with the complement system. The first is going to be the first pathway is going to be the opsonization, which occurs when excess C3b binds to bacteria. The C3b protein binding to the bacteria allows the bacteria to be opsonized, meaning it's just being prepared to eat, which this just allows the bacteria to be phagocytized a lot easier and just allows it to actually be taken out of the body. The second outcome is the inflammatory response, which occurs when the C5a protein is activated and starts attracting phagocytes to where the complement system was activated. The C3a and C35a proteins can induce changes in the endothelial cells of blood vessels lining and increasing permeability, which pretty much just turns into inflammation of the area just because you're having all that fluid leak out of the blood vessels. The final outcome is the lysis of foreign cells. This occurs when many different proteins such as C5b, C6, C7, C8, and C9 all spontaneously assemble in cell membranes and form structures called the MACs, or the membrane attack complexes. These forms in the pores of the membranes of the cells, causing them to lyse just because it has all that space. It just takes all of that space of those spores up, so it just has no room to grow, causing it to lyse. MACs have a stronger effect against gram-negative bacteria just because they have a thinner peptidoglycan layer. And finally, the regulation of the complement system. The complement system is regulated by the fact that the host cell has the complement regulatory protein. This protein allows the C3 to bind and then inactivates the protein, therefore just regulating the complement system. The Christian worldview is going to be really simple. First, I want to talk about the fall and disease. So, in the Christian theology or worldview, disease and decay entered the world through the fall. This is seen in Genesis 3. The pathogenic microbes and immune system malfunctions, like autoimmune diseases, all this can be seen as like a result of broken and creation, again from the fall. And the immune system demonstrates wisdom and design that points to an intelligent creator, which kind of points us towards God in that aspect, just because everything's made through his image. Its ability to recognize the self from non-self, remember past infections, and coordinate millions of cells in harmony is a marvel of divine engineering. And also just an example of this, the adaptive immune system like remembers pathogens similar to how like humans remembered lessons through experience. This memory ensures faster, stronger protection, next time, showing foresight, purpose and design, all of that. So just kind of like a biblical connection to that. It's going to be seen in Psalm 147, verses 3 through 4. Says, He heals the brokenhearted and binds up their wounds. He determines the number of the stars and calls them each by name. So this kind of just goes along with the bacteria and cells in our body. Everything was made by God and in His purpose and in His image. So kind of just as God knows all the stars, for like in this verse, He designed and recognized each cell of the body, to be able to heal itself, to be able to fight off these bacteria.
What is fever? What does it mean when your body temperature rises? Fever is a rise in the body temperature above normal. It usually happens in response to infection or inflammation, and it's the body's natural way of fighting germs, meaning when people get sick, we all get fevers, and we see it as a normal thing because it happens almost to everyone who is sick, and then we just cure it with a little rag or medication. And it's just a common thing that happens to people when people are sick. How fever works. First is the slow germ growth. So many bacteria and viruses reproduce more slowly at higher temperatures, giving your body a better chance to fight them. Then the boosts immune response. So the fever activates white blood cells, helping them fight and destroy germs more effectively that are causing the fever. And then we enhances chemical signals. So fever increases the release of immune chemicals that coordinate the body's defense. And then signal rest and recovery. So higher body temperature encourages rest, converting energy so that the body can focus on healing, which is why a lot of people who are sick and have fevers tend to lay in bed and just rest up and let the body rest and fight the infection. Fever and the complement system, they correlate because fever slows pathogens, giving complement proteins more time to attack. They also complement proteins trigger inflammation, which can contribute to fever. And together they form a coordinated defense system. So fever strengthens the immune system while the complement system destroys the pathogens. So together they work together to fight off the fever and help each other throughout the whole process.
Hi, my name is Theone Divina Gracia, and I'll be talking about the types of antibodies and outcomes of antigen antibody binding. First, what are antibodies? Antibodies are Y-shaped structured proteins that are created by our immune systems so that they are able to fight off unwanted substances that enter our bodies, like antigens. So that means if a virus, infection, or parasite enters into our body, the antibodies will help fight it off and destroy it so that we feel better. They're also known as immunoglobulins, and they are produced by B cells. As we have talked about many times in lecture, structure equals function. Whether it's a cell, organ, or molecule, how it is structured helps with its function. The structure of the antibody is Y-shaped. They're also known as antibody monomers. They consist of heavy and light chains linked by disulfide bonds. On the top left region of the photo shown, we have the Fab region, and the Fab region is seen on both arms, and they are the region that binds the antigen to the antibody. Within the Fab region, we have the variable region, and that region recognizes the antigen and what type of antigen it could be. On the bottom, the base of the antibody, we have the Fc region, which triggers the immune and complement system. And that basically means when the parasite or the antigen or the virus enters into our body, the Fc region tells our body what to do and how to fight it off. And within the Fc region, we have the constant region, and that region recognizes the type of antibody class, which we will talk about in the next few slides. But since we understand the structure, now we can understand the function of antibodies and how our bodies recognize and bind antigens, since we have learned now about the structure. So how does our body recognize and bind antigens? The structure of the antibody monomer helps in recognizing and binding antigens. The variable region found on the arms of the antibody function is used to identify the type of antigen, while the constant region recognizes what type of antibody is needed to fight it off. The lock and key method is used to bind antigen to antibody. Antigens are the key, while the antibodies are the lock. They both have specific structures relating to one another. Remember how we talked about how the constant region and how it looks for the types of antibodies it needs to fight off an antigen? Well, here are a few of those different types of antibodies. First, we have IgG. IgG is the most abundant and longest term of protection. It is the only antibody that can cross through placenta, which means it can protect a fetus against infections. Second, we have IgE, which is found within mast cells or basophils, and they release histamine. It helps in destroying parasites and aiding in allergies. Third, we have IgD. IgD aids in making and growing of antibodies. They can be found on B cells. Fourth, we have IgM, which is the first type of antibody class produced. They are pentameter shaped, consisting of 5 antibody monomers. Again, structure equals function. So the structure of the IgM antibody, which is pentameter shaped, helps in destroying antigens. They control infections in bloodstreams. And lastly, we have IgA, which are found in secretions. They create a dimer, which makes 2 antibody monomers join, creating 4 antigen binding sites. They are found in breast milk, which helps against infections in babies. Here are the outcomes of antigen to antibody binding. We have 6 of them, and the first 3 we'll be talking about is neutralization, opsonization, and complement system activation. Neutralization is when an antibody molecule layers over a virus, which results in that virus not being able to attach itself to an organism cell and harming it. Opsonization, a phagocyte cell is able to engulf object easier due to their cells having receptors on the Fc region, which helps marking an antigen easier. Third, we have complement system activation. Complement system proteins create a pathway to produce opsonate C3b to help destroy and attack the virus. Fourth, we have immobilization and prevention of adherence. Antibodies attach to flagella or pili to prevent bacteria from moving and attaching to organisms and destroying them. Fifth, we have cross-linking. The arms of the antibody monomer grab multiple antigens that are similar, resulting in a big antigen to antibody complex so that phagocytic cells are able to engulf them. Lastly, we have antibody-dependent cellular cytotoxicity. Natural killer cells will attack antigen since antigen is being attacked by multiple antibodies, making it a big target. Here we have our references that we used. And thank you for listening to our slide presentation.